Yes, I'm here. Can you hear me? Can you hear me? Allison, I'm having trouble with the
Yep.
connection. Are you there?
Yeah, you're fine.
Okay.
Just cutting out.
So a couple of disclaimers before we start. I'm just getting over a nasty virus, and so if I have to stop and cough, please excuse the interruptions. In terms of financial disclosures, I have been a principal investigator for 2 of the 3 products that we're going to talk about today, CAR T-cell products. I've turned those responsibilities over to another investigator at my institution recently. I also do some consulting for Novartis. I have not received any financial compensation for that, and I do receive some other research funding for Novartis for some other drugs that are currently being investigated in clinical trials. I don't own any stocks, so I really don't have any financial conflicts. So here we go. The objectives for our session today is that I hope by the end of this presentation you'll be familiar with what CAR T cells are, how they work. I'm going to summarize the 3 CAR T cell products that are presently available for childhood ALL in North America. Hopefully soon the 3 of those will be available in Canada. I'll talk a little bit about, in addition to how CAR T cells work, I'll show you some of the data that's available about how effective they are. We'll talk a little bit about side effects, and then we'll end with a little bit of looking into the future about where CAR T-cell therapy may be headed. So for those of you who are not familiar with childhood ALL, I'll just have a little bit of background information. ALL, or acute lymphocytic leukemia, is the most common form of cancer in children. It represents about 25% of all cases of childhood cancer. It has 2 main subtypes, B lineage ALL, which represents about 85% of childhood ALL, and T lineage ALL. And for reasons that we'll talk about shortly, CAR T cells are only relevant, only applicable to patients with B lineage ALL. They won't work in patients with T ALL at the present time. And acute lymphocytic leukemia arises in normal blood-forming cells. The medical term for that are hematopoietic stem cells. So if you look at this figure that I have here, all blood cells arrive from a mother stem cell, if you will, a progenitor cell. And the first decision that they have to make is, are they going to become a myeloid stem cell and make things like neutrophils and eosinophils, or are they going to become a lymphocyte? The earliest form of a normal healthy lymphocyte, the least mature form, is called a lymphoblast. Some people may think that because the disease is called acute lymphoblastic leukemia, that it may mean that all lymphoblasts are malignant, but that's not the case. We all have normal lymphoblasts as a part of our hematopoietic system. And then the lymphoblasts will further mature or differentiate to become B lymphocytes or T lymphocytes or natural killer cells. So all of these cells are types of white blood cells. The progenitor cells also give rise to red blood cells and platelets. So in terms of how we're doing currently in childhood ALL, the happy news is that most children with ALL who are treated on protocols that are used in Canada, COG protocols or Dana-Farber protocols, are cured the first time around with standard intensive chemotherapy. These are data from the Children's Oncology Group showing that the overall survival for children with ALL is now predicted to be more than 90% with the current generation of studies. And these are very similar data from the Dana-Farber group showing the success rates with their protocols. So Dana-Farber protocols are used by the Children's Cancer Programs in Quebec and also one program in Ontario. The remainder of Canada, the treating centers use Children's Oncology Group protocols. So regardless of which approach is used, the overall survival is really excellent, about 90%. So if we're doing so great with childhood ALL, why do we even need to talk about these miraculous new therapies? Well, the reason for that is that with these current therapies, about 15 to 20% of children with ALL will relapse. Overall survival, the statistics I showed you, include patients who are alive regardless of whether their disease has recurred or not. So 15 to 20% of children will still relapse. Our current approach to treating those children is to give them, following a relapse, very intensive chemotherapy. And depending on certain factors that we look at, many but not all of those patients would be recommended to then undergo an allogeneic bone marrow transplant. Allogeneic meaning that it would be bone marrow donated by someone else. The survival rates for this approach are, in some studies, about 50%, and other studies are less. And so we really want to do better. And if after receiving that treatment, including a bone marrow transplant, a patient has another relapse, then it's much more difficult to cure patients. And because childhood ALL is the most common cancer, even though it's only a relatively small percentage that will relapse, the burden of childhood ALL in relapse is enormous for our patients and for society. In fact, one of my mentors told me many years ago that if childhood relapsed ALL were its own disease, it would be the fourth most common type of childhood cancer. So that may put into perspective for you the reason that we need new therapies. This is a slide provided by a colleague in Germany, Dr. Pfister. This is sometimes called the Pfistergram, and it just shows that survival after relapse for many childhood cancers is unacceptably low. The two figures here in the upper left show what happens with patients with high-risk ALL after relapse. Regardless of what treatment they receive, you can see that the cure rate is less than 20%. Similarly, children who undergo a stem cell transplant or bone marrow transplant for ALL, if the ALL comes back after that, the survival rates here you can see perhaps are less than 10%. So enormous challenges in those settings. These are some data from investigators at a couple of institutions in North America, including sick kids, again showing that patients who have a hematologic malignancy and undergo a bone marrow transplant, which I think many patients and families look at as curative therapy, and historically has been the only curative therapy for many patients who have a relapse. If you have the bone marrow transplant and despite that the leukemia comes back, the overall survival is really quite low, about 15%. So we need better treatments. And so for a number of years, scientists have been working on harnessing the immune system as a new approach to treating cancer. Currently, we use chemotherapy, which is essentially controlled poisoning. We can use radiation therapy in selected cases to supplement chemotherapy. Now we're entering a new era where we have completely new types of tools to treat cancers, including childhood ALL. And immunotherapy is going to be, I think, one of the most important tools in our armamentarium. So in 2013, about 5 years ago, Science magazine recognized cancer immunotherapy as it was beginning to enter the clinic as its scientific breakthrough of the year. Now, there are many different types of cancer immunotherapy, and we're not going to talk about, we're only going to talk about really CAR T-cells today. But I would just point out that there have been for a number of years attempts to vaccinate patients against cancer with mixed levels of success. There's a very exciting category of immunotherapies called checkpoint inhibitors, which are now being used widely in some of the most common adult cancers, and many of those drugs are now approved by the FDA and Health Canada and are commercially available. They're continuing to be investigated in clinical trials in terms of being given in new combinations, but they are now a part of the standard armamentarium in adults with cancer. The pediatric trials, as always, are lagging behind and are really just getting underway. Another class of immunotherapies are drugs called immunotoxins. And because this is an audience, I believe, that's interested in childhood ALL, one of those drugs is a drug called inotuzumab, which is an antibody that's linked to a toxin. It's being investigated currently in children. It's not yet approved in children, although it is approved in adults with ALL. The early data look very promising, and I expect that in the near future, inotuzumab will become another approved drug that will be available for us to treat children with recurrent disease. And then the fourth and final category or type of cancer immunotherapy for childhood ALL are so-called T-cell engaging immunotherapies, immunotherapies that work by harnessing a patient's own T-cells in different ways. The first of those drugs was something that's not a CAR T-cell, although there are some similarities. It's a drug called blinatumomab, and I think this drug is now available across Canada, both in the context of clinical trials and as an approved product within the last year. It is a class of drugs called biphenotypic T-cell engagers, or BiTEs for short, and it's a type of antibody that works by bringing together T-cells, which express CD3, a protein on their surface, and then also B lineage leukemia cells, virtually all of which express a different protein, something called CD19 on their surface. And we're going to talk more about CD19 momentarily. And then really the topic of today's discussion are CAR T cells. So what does CAR T cell mean? As you heard from Allison, the CAR stands for Chimeric Antigen Receptor T-cells. And you'll remember from your mythology lessons that a chimera is a multi-headed beast of some sort. And that's really what these CAR T-cells are. So if we look at this cartoon, we can see on the left here something called MAB. That's short for a monoclonal antibody. Antibodies are proteins made by our immune system to help recognize infections and other potentially dangerous agents. And they have two ends. One end is specific for recognizing a specific protein. That's called the variable fragment, or FV here for short. And it's now possible to make antibodies in the laboratory so that they all recognize exactly the same thing. And that's called the monoclonal antibody. And that's what we see here. So monoclonal antibodies are actually used to treat lymphoma and other types of cancer themselves. That's yet another type of immunotherapy. But against childhood ALL, they're not very potent at all. So in this approach of making a chimera, various parties have taken this variable fragment that recognizes a protein, and specifically here, it recognizes CD19, a protein that's expressed on the surface of B cells and very little else. And then it's attached to a T-cell signaling domain. So this is not this is a chimeric antigen receptor. This is not something that would occur in nature. This is something that's been constructed through genetic reengineering. So this chimeric antigen receptor is a chunk of DNA that's packaged in a retrovirus, a type of virus that will be able to inject DNA into the cell that it targets. And this retrovirus then can insert this DNA into a patient's T-cell. The DNA will be transcribed into protein, and this chimeric antigen receptor, or CAR, will then be expressed on the surface of the patient's cell, so that it is like a receptor that will recognize the leukemia cells. This is the genetic engineering that, or gene therapy, that distinguishes CAR T cells from normal T cells, which would express here what is called the T-cell receptor, but is not really specifically targeted to a patient's leukemia cells. Now, retroviruses include a whole class of types of virus. One type of retrovirus is the human immunodeficiency virus, or HIV. And so you may have read in some of the lay literature that CAR T cells are using HIV. Strictly speaking, that's not true. Broadly speaking, I think it is correct to say that that viruses like HIV are being used to facilitate the gene therapy, to introduce this foreign genetic element of the chimeric antigen receptor into the T cell. So there are many different types of proteins expressed on the surface of B lymphocytes, B lymphocytic leukemias, in really all cells. That's how cells recognize each other and understand their jobs. Why was CD19 picked by not one but now 3 different organizations as the desired or preferred target for childhood B lineage ALL? So the reasons are that this is an antigen on the surface of the B ALL cells that really doesn't occur on much else. It occurs on normal B lineage lymphocytes, and then it occurs on the B leukemia cells that are essentially a normal healthy B lymphocyte that has gone bad. So it's not going to stick to many other things, and that will reduce some of the potential side effects. It's expressed on almost all developmental stages of B cells. If you look here, you can see this black line. We have the earliest B lymphocytes on the left, the stem cells, all the way on to the right, the most mature B cells, plasma cells, and CD19 is expressed at many stages. ALL cells are usually relatively immature, but virtually all ALL cells still have CD19 expression. This is chronic lymphocytic leukemia. This is a type of lymphoma that occurs primarily in adults. And because they also express CD19, then CAR T cells that are directed against CD19 could also be used to treat those diseases. And in fact, we're seeing that in adults. And so the fact that CD19 is expressed on virtually all B lineage ALLs means that this is a drug that can be used for virtually any child with B lineage ALL who relapses. So there are currently 3 CAR T cells that are targeting this CD19 that are undergoing trials in North America for childhood ALL that are either in or will be coming to Canada. There are actually many other CAR T cells that are in development, but these are the 3 that we expect to either currently have access to in Canada or expect to be able to have access to in the very near future. And I'll talk about each of these. I'm going to start with the CAR T cell whose medicinal name is CTL-019, the 019 referring to the CD19 protein target. And these are the cells that were developed academically by the Children's Hospital of Philadelphia, usually known as CHOP. And the rights to this therapy and the commercial development has then subsequently been by Novartis. I'm going to start with this drug because this is the first and so far only CAR T cell that has been approved to treat childhood ALL by the FDA. This is the FDA's announcement on their website in August 30th of last year. This was the first CAR T cell therapy. It's actually the first gene therapy that's been approved to be administered to humans. So this was a very big deal. Here's a little bit more information on that same day from the New York Times. FDA approves first gene-altering leukemia treatment, and they note the cost of the drug, quite expensive. This is what a bag of these CAR T cells that have been genetically engineered look like. Here's the now FDA-approved name, tisagenlecleucel, which is why most of us still call it CTL-019. The brand name is Chimera, and this is a picture of the plant that Novartis has in New Jersey where these cells are actually manufactured. So a little bit more about this particular flavor of CAR T cell and what makes it different from at least one of the other products that are available. So we've talked about the variable regions that come from monoclonal antibodies that target the CD19. These are on the outside surface of the CAR T cell, this blue horizontal line that you see here. On the inside of the CAR T cell are the signaling domains, and these are these normally occur in CAR T cells, although they're not hooked usually to the CD19 domain. And these are the cells that internally, once they're activated by the CD19 locking up with this receptor, they tell the T cells to grow, to proliferate, to increase in number, and to go do their job, which is to kill the target. Here, the CD19 expressing leukemia cells. Not all, well, all CAR T cells have some sort of signaling domain. Not all CAR T cells have this particular co-stimulatory domain called 4-1BB. This may be very important in CAR T cell therapy in terms of optimizing how effective it is, although we're still waiting for a lot of the data to emerge with longer follow-up, because it's believed that this 4-1BB co-stimulatory domain will cause the CAR T cells to persist in the patient for a much longer period of time than if that's not present. How important that is, I think, is not yet clear. So let's look at the logistics of how CAR T cells work. So I've explained to you what a CAR T cell is, how it's manufactured by taking this genetically engineered DNA construct using a viral element to transfer it into a patient's T cells and then to be expressed on the surface. But how do we actually get these drugs to our patients? How do we get their own CAR T cells out in the first place? And so that's what the purpose of this slide is. And these slides I'm showing you now and many that will follow are in the public domain. These are information provided by Novartis as a part of their FDA approval process, and so they're publicly available and in the public domain. This is not confidential information that I'm presenting. So here's a patient with relapsed B lineage ALL. At the present time, the clinical trials have focused on a particularly high-risk group of patients with B lineage ALL. So those are not all patients with B lineage ALL, but only patients who've relapsed. And specifically, it does not include patients with their first relapse, because we have treatments, even though they can be brutal, intensive chemotherapy and bone marrow transplantation. With these treatments, we can still cure a reasonable percentage of children with first relapse of B lineage ALL. So CAR T cells are being investigated in the worst of the worst cases. So these are for patients who've had a second or greater relapse of their ALL, or patients with ALL that has relapsed after a bone marrow transplant. We saw in a slide earlier that 15% or less of those patients will survive. There is no true effective therapy in that setting. Or a very small percentage of patients with so-called refractory ALL, which means their leukemias never went into remission to begin with. So those are the patients that are in the current studies, and that is the approved indication. So highly resistant or refractory pediatric ALL. So the first step is to have such a patient, confirm that they have a relapse, and then to collect their white blood cells, which will include their own healthy normal T cells. So these are the patient's own immune cells that work, but they haven't been educated or engineered to be able to specifically attack the leukemia cells. So they're removed by a process called leukapheresis or apheresis. That's actually a process that has been used for a number of years in children and adults with various types of cancers to collect their own white blood cells, usually before something called an autologous bone marrow transplant, where they will receive high doses of chemotherapy and radiation in an effort to wipe out the leukemia or the other whatever type of cancer is being treated, with doses of chemotherapy and radiation that are high enough to try and kill the cancer, but which will certainly wipe out their normal healthy bone marrow. And then after that treatment is given and is out of their system, these cells that have been collected and stored outside the body will be given back to the patient to reseed the garden, if you will, allow their bone marrow function to be restored. But here, rather than just going into a freezer and then being given back to the patients later, these cells are then put on a plane and they are sent to a manufacturing facility where they are processed. And so you can see the various steps here. They are enriched to try and get as many of the T cells, which are what we want to do the genetic engineering for, and as few of the other cells as possible. Then they are put together with the lentivirus, represented here by the small orange particles, and some of those will introduce the chimeric antigen receptor into the T cells, and the protein will end up being expressed. And these are the cells that we really want, so they're purified to maximize these transduced cells. Those cells are then grown up outside the body through a process called expansion to increase their numbers, and then they undergo a number of quality checks. They're put back on a plane, and they come back to the clinical site. For this study, as we'll talk about more in a moment, the two sites that have been and continue to participate in clinical trials of CTL-019 are the Hospital for Sick Children here in Toronto and Saint-Justine Medical Center in Montreal. These cells are then administered to the patient and allowed to do their work. They're infused just like a bone marrow transfusion or a red blood cell transfusion, and those CAR T cells then in the bloodstream can go to the bone marrow, the lymph nodes, go everywhere, seek out the leukemia cells, and use this CD19 receptor to look for and stick to leukemia cells. And because CD19 is expressed on almost all B lineage leukemias in children, but on very little else, that's really all they're going to stick to. So on the left, we see a patient's T cells here after they've undergone this genetic engineering, and you'll see the purple receptors here are native CAR T cells that would be CAR T cell receptors, native T cell receptors that would be present on any T cell. And then these orange to red receptors are the chimeric antigen receptor construct. These are the things that will stick to CD19. On the right here, we have a leukemia cell that expresses CD19. And so this protein will be recognized by this receptor. They will connect, and that will then trigger the CAR T cell to do two things. Number one, to release cytokines that will contribute to cell death and also cause the cells to proliferate or to grow. And so part of the success of CAR T cells is that this signaling is very potent, and the CAR T cells can multiply many-fold, and that seems to be important in how effective they are. So the trial that was conducted, sponsored by Novartis, was conducted around the world, a global phase 2 study of CTL-019 in children, adolescents, and young adults with relapsed or refractory ALL, as we talked about. It has a number that you see listed here. Novartis, the sponsor, called it the Eliana trial. And so there were 2 sites in Canada, as I mentioned, 13 sites in the United States. There were a number of European countries that participated, at least 1 site. Japan had 1 site, and Tokyo, I believe, and Australia had 1 site. So this truly was a global site. You can see the number of patients enrolled in each country that are listed here. And so this is really, I think, one of the most important slides in the presentation, because this shows the results of the CAR T cell therapy in this group of highly resistant children with relapsed refractory ALL after receiving CAR T cells. So they had an overall survival. That means the children are alive either with or without a recurrence of their disease at various time points after getting the treatment. This is a group of patients that would be predicted to have a very poor survival. And you can see that at 6 months after the treatment, 90% of the children were still alive. 6 months, we go up here to the percentage, it would come out to be about 90%. And at 12 months, a time when most children with any other treatment would have, the majority of patients would have had a recurrence or of their disease and not be surviving. at 12 months, 80% of the patients are still alive. So this was really an unprecedented result, and led the FDA to provide approval for this drug without requiring a randomized trial, because I think it was likely felt that it would really be unethical, and that no one would agree to who could get CAR T cells wouldn't be willing to do that. So that's the first drug, and so far that's the only drug that's been approved by the FDA or anyone for treating children with ALL. Now I'm going to tell you a little bit about a second product called KTE-C19. This is a drug that was developed and initially studied at the National Institutes of Health in Bethesda, Maryland, a part of the National Cancer Institute of the U.S. The rights to this drug were then subsequently purchased by a company called Kite, a relatively new small company focused on cellular therapies, and then Kite was subsequently bought by a much larger drug company called Gilead. So the people at Kite made a decision, because Novartis was studying children with relapsed CD19 positive ALL, and because their drug, as implied by the name, also targets CD19, they decided to study a different population of patients whose tumors also express CD19. They decided to study adults with large B-cell non-Hodgkin's lymphoma who had relapsed or were refractory to treatment after at least 2 lines of therapy. Another very high-risk group of patients for which there's no clear effective therapy. And what they saw with this different flavor of CAR T cells were also really unprecedented responses. Over 100 patients treated. The overall response rate was over 80%. 80% of the patients were still alive at 6 months. And so the FDA approved this drug for adults with large B-cell non-Hodgkin's lymphoma who had failed multiple prior therapies. I can tell you that this drug, the brand name of which is now Yescarta, is undergoing study in children. In fact, at the Hospital for Sick Children, we have a trial for children with relapsed refractory ALL of this type of CAR T cell. But because that study is still ongoing, there are no published or publicly available data about how well it does. There are some data from a much smaller study done initially at the NIH, and this drug appears to be highly effective, this flavor of CAR T cells. However, it does not seem to persist or be sustained in the patient's immune systems for long periods of time, because this one does not include that 4-1BB co-stimulatory domain. And so in the study from the NIH, children who received this treatment had a very high chance of going back into a very deep remission, but only the children who subsequently underwent a bone marrow transplant, whether they'd had one or not before, only those children who underwent a bone marrow transplant remained in remission. If you didn't get a bone marrow transplant after receiving this type of CAR T cells, the leukemia always came back. I will point out that's very different from the experience in adults with lymphoma. And although many of these patients with lymphoma did not get any treatment after the CAR T cells, they seem to have sustained remission. So there's still a lot to learn about CAR T cells and how they may work differently in adults versus children, and lymphoma versus leukemia, in terms of what's the optimal makeup of the vector. This KTE-C19 is still in ongoing investigations in children with ALL, and hopefully we'll know more about that soon. The third drug that we expect will be coming to Canada soon is being developed at Seattle Children's Hospital. They've done some preliminary studies at their hospital. The response rates look very good. It seems to be safe and promising. And BC Children's is hoping to be able to open a trial of the drug developed at Seattle Children's Hospital that would be available to children in British Columbia. Those arrangements are not finalized, but they seem to be moving along well. And so at least for children in BC, this product may be available. This is from the Seattle Children's Hospital website. And although none of their products are yet approved by the FDA, they have very promising data, and they are moving on already to build the next generation of CAR T cells that will target not just CD19, but actually CD19 and another protein on the surface of ALL cells, which at the same time, which may make them work even better. So these therapies sound great. If they are so effective, why would we not quickly move to giving them to all children with ALL and replace the poisons, the chemotherapies that we have to use now? Well, we have a lot to learn about these drugs, but we do know that they can have serious side effects. They're very different than the side effects of standard chemotherapy, but they are significant nonetheless. We're going to talk about two of these, cytokine release syndrome and neurotoxicity. So cytokine release syndrome has this name because the T cells, the CAR T cells that get activated through the process I described, one of the things they do is they release signaling proteins called cytokines over the course of the several days as they begin to grow in a patient. And cytokines are the proteins that cause a flu-like or sepsis-like syndrome. If I share these symptoms with you, you should recognize them if you've ever had the flu. Fever, chills, low blood pressure, respiratory issues. If we do blood tests, we can see that the liver gets irritated. We can see that there can be clotting problems. This can be mild. Some patients don't even get any cytokine release syndrome that's clinically evident, although if we measure cytokine levels in their blood, we'll see some evidence of this. In some patients, it can be very serious. The blood pressure can drop so low that even giving fluid boluses is not sufficient, and the patients have to go to the ICU to get support medications to keep their blood pressure up. Similarly, most patients will not develop respiratory issues, but it can be that patients will need supplemental oxygen or even ventilatory support. And these are potentially life-threatening if they don't reverse. This cytokine release occurs after blinatumomab, which activates T cells in a different way than CARs, as well as CAR T cells. Blinatumomab, which became available before CAR T cells did. This was the first time this syndrome was recognized. Those patients get dexamethasone, which, as you may know, is a very potent steroid that will suppress the immune system. When the first patient to receive CAR T cells at CHOP was treated, she developed severe cytokine release syndrome, was given dexamethasone. It didn't work. And so a very smart doctor there I know gave looked at her cytokine profiles, saw that her IL-6 level in her blood was very elevated, said, we have a drug for that, and went to the rheumatology program and got a drug called tocilizumab that's given for severe arthritis. And she received this drug, and within 24 hours, she was off the ventilator and off blood pressure medications, and significantly better. And so this has really become a very important treatment for cytokine release syndrome, this drug called tocilizumab. And in a very forward-thinking way, on the same day that the FDA approved CAR T cells, they also approved tocilizumab, which was not an experimental drug, but they approved it for treating patients who develop cytokine release syndrome. So now not only the drug, but the antidote to some of the side effects are approved by the FDA. This is an algorithm about how to manage patients with cytokine release syndrome. I'll skip over this in the interest of time. And I'll also mention that the second side effect that we see after giving CAR T cells is neurotoxicity. And again, this happens with blinatumomab, which also targets CD19. The neurologic symptoms can not occur at all. If they do occur, they can be mild. Patients may have handwriting that looks a little sketchy, so to speak. It can be serious. It can be, the first symptom can be a seizure. Why this happens is unknown, despite many studies looking into potential causes. Most, if not all, of these patients end up getting a brain MRI shortly after a neurologic event, and there's no evidence of swelling or other changes that are seen. And so far to date, all of these neurotoxicities have been fully reversible. So this is a very scary symptom. It's potentially life-threatening, but at least in the CTL-019 trial, there was no patient who died of a neurologic toxicity caused by the CAR T cells. And we'll skip this in the interest of time. So approaching the end here, is CAR T-cell therapy curative? Well, what we have all been told, and I think was the expectation, was that CAR T-cells might be very good for putting patients back into remission, but we didn't expect them to lead to long-term cures. So so-called bridge to patients being able to get a hematopoietic stem cell transplant or bone marrow transplant. However, it turns out that the flavors or types of CAR T-cells that have this 4-1BB co-stimulatory element seem to persist for very long periods of time in some patients, not in all patients. Why, we don't know. And if those patients have CAR T-cells that hang around in a patient's body, and after all, they are the patient's own CAR T-cells, the body would not recognize those T-cells as foreign because they came from the patient, although they have been genetically engineered with a foreign protein. But if they stay around, these CAR T-cells, in the patient's body, they can continue to pick off leukemia cells as they arise, and that may mean that patients may be in remission for a very long time, may be cured. Too early to say. The first patient treated with CAR T-cells that I spoke about earlier at CHOP is a girl named Emily Whitehead, and I can share this information with you because she has her own website and her own YouTube videos and all of these sorts of things. She remains in remission for more than 5 years after she got CAR T-cells treatment and no treatment after that. She didn't have a second bone marrow transplant. She'd already had that. The doctors rightly told her, we don't have any proven treatment that can cure you. You've had them all. We have this new experiment, CAR T-cells, and Emily went through a very difficult time. She is now 5 years cancer-free, remains in remission with no evidence of leukemia. And so fortunately, I think for the whole childhood ALL community, the very first patient who got this treatment has done very well and has become an inspiration to all of the other children who would benefit from this. So my last slide, and then we'll be finished, is to talk about sort of where we are in the current landscape in Canada and where we're headed. So the first product I talked about, the Novartis CTL-019, is approved by the FDA in the United States for childhood ALL, and it's commercially available. Centers can buy that drug and give it to their patients. In Canada, Health Canada has undertaken the review, but we don't expect to receive word about whether this is going to be approved in Canada until late in 2018. I think everyone expects that since it was approved by the FDA and since Canada was a part of these studies, that it will be approved here as well, but we'll just have to wait and see. The Kite drug that I mentioned is the second one, is still undergoing its efficacy trial, so I think it's going to be another 6 to 12 to 24 months before we have sufficient data about it to say how safe and effective it is. And then the Seattle team is lagging a little behind, but they're quickly catching up. And we know that we hope that our colleagues at BC Children's in Vancouver will be able to provide access to that particular drug for their patients. So some other things that are in development. CD19 is a great target. It turns out that one of the ways that leukemia cells can outsmart CAR T cells is that they can figure out ways to stop expressing CD19 on their surface. And if that protein's not there, then there's nothing for the CAR T cells to stick to. So people are developing CAR T cells that target other antigens. Here, CD22, something that's also expressed on many but not all childhood ALL cells. I shared with you that Seattle is about to open a trial, as are some other groups, that target both CD19 and 22 with a single CAR T cell. Maybe that'll avoid some of these ways that CAR T cells outsmart, that the leukemia cells outsmart CAR T cells, but we'll have to wait and see. This therapy could also work for acute myeloid leukemia, less common in children than ALL, more common than ALL in adults. But those are the cells that, when they are healthy, become neutrophils, and you need your neutrophils to avoid getting infections. So if you give a patient CAR T cells and they take out both the leukemia cells and the healthy neutrophils, they're inevitably going to get an infection and die. So either those patients have to quickly go to a bone marrow transplant, or there has to be a way to let the CAR T cell do its job and then trigger a suicide switch that will kill the CAR T cells off after they've gotten rid of the leukemia. Both those approaches are being investigated, but it's too early to say really about that. And then, of course, if they work for leukemia, maybe they could work against other types of cancer as well. And so they're both these biphenotypic T-cell engagers and CAR T-cells that are being evaluated for many of the common cancers of children and adults. But it's not clear whether they're going to work or not, because it may be that having the leukemia cells immediately accessible in the blood rather than in a big mass that may not have great perfusion. We don't know how important those factors are, and the only way we can learn are to conduct more clinical trials and see what happens. So that really completes my presentation. I'm going to stop there and ask Allison if we have time for a couple of questions. Thank you.
Thank you so much, Dr. Whitlock. How's my audio now? Are we doing okay?
Yes, I can hear you just fine, thanks.
Perfect. So apologies to everyone for my audio kind of cutting out earlier. It was a computer issue, which I've now resolved. So we'll just remind everybody real quick, if you do have a question, there is some time here. You can ask it by typing it into the window on your right, and we'll address as many as we can. We have one question here so far, so I'll read it out to you. So reports by ICER and NICE in the UK suggest that some refractory ALL patients, who respond to CAR T-cell therapy go on to also receive SCT. Are CAR T-cells not curative, and what will make stem cell transfusion necessary after CAR T-cell therapy?
So, yeah, we did touch on some of those issues later in the presentation. So CAR T-cells for ALL can be curative without a subsequent bone marrow transplant, but that may depend on how they're made. So the Novartis product, which includes the 4-1BB element, they can persist for many years, and those patients do not appear to necessarily require hematopoietic stem cell transplant. We don't know at the time of the CAR T-cell infusion, we don't know how to predict whose CAR T-cells will persist and whose CAR T-cells are going to eventually go away. So it's sort of a wait-and-see approach. We do know that for the Kite or the Gilead product that was developed at the National Institutes of Health, that it does not persist, it does not include the 4-1BB, and that in the earliest studies from the NIH, that patients who got that CAR T-cell needed to go on to get a bone marrow transplant in order to have a sustained cure. I should also point out that CAR T-cells, one of the situations in which they're being used are in patients who've already had a bone marrow transplant, and not every patient is able to undergo a second bone marrow transplant. So in that situation, having a CAR T-cell that would persist might be important in order to try and avoid a second bone marrow transplant, which can be very toxic. Are there any other questions?
Okay, thanks so much for that question. Yeah, we have just two more coming through here, so I'll read this next one. It says, is the manufacturer the only one that can genetically engineer the T-cells, or is it possible to do in centers in Canada to reduce costs?
So that's a great question. So I would point out that both the CTL-019 that's now being commercialized by Novartis, that was developed initially at an academic medical center, the Children's Hospital of Philadelphia and the University of Pennsylvania jointly. And the second product that's being tested broadly, the Kite or Gilead product, was initially developed in an academic center at the National Institutes of Health. So yes, it is possible, entirely feasible, that CAR T-cells can be developed and produced at academic centers, including academic centers in Canada. And there are a couple of efforts afoot to do that. We aspire to be able to do that here at Sick Kids. There's also a group, BioCanRx, that is working to develop made in Canada CAR T-cells as well. How we're going to do that, whether we're going to develop these things from scratch or work with existing producers to buy the rights to use their constructs and then do our own transfections here in Canada, all of those things are in the process of being explored and worked out. But I think in the long term, yes, we will be able to make CAR T-cells here in Canada, and hopefully that will reduce the cost.
Okay, great question. Thank you. And our next question says, based on the current evidence base, is there an evidence to suggest that a second infusion of CAR T followed by pembrolizumab, pardon my pronunciation, is comparable or superior to other alternative options?
Yeah. So as I mentioned earlier, patients who receive CAR T cells, some patients may have prolonged existence of those CAR T cells or persistence in their bloodstreams. Other patients may have the CAR T cells be around for a shorter period of time and they eventually disappear. Why that happens is not clear, but there's some early data from CHOP suggesting that some patients can develop an immune response to the CAR T cell protein that expressed on the surface of the patient's T cells. So the T cells are the patient's cells and they're not going to be rejected. But some patients may have enough immunity that they recognize this foreign protein, the genetically engineered protein that ends up on the surface of the CAR T cells as a foreign protein, develop immunity to it, and that's what gets rid or causes the CAR T cells to go away. So giving that same construct back has worked in some times, and in some patients it's not. Investigators are looking at ways to modify the CAR T cells to reduce or hopefully even eliminate the possibility of developing immunity to that expressed protein by fully humanizing the CAR T cells. And there's some early trials underway by a couple of groups to do that. So those would be the next generation of clinical trials that we would hope to do. And then there is some evidence that activating the immune, so some patients have a very good response and go into remission, but some patients do not. And there is some very early evidence that giving checkpoint inhibitors, which activate the immune system in a different way, when combined with CAR T cells, may allow some patients to have a response when they don't respond just to CAR T cells by themselves. But this is very tricky business because checkpoint inhibitors, we don't really have control over how much immune system activation we get. And what we know from the adults that have been treated with checkpoint inhibitors, even without CAR T cells, that some of those side effects can be serious, life-threatening, or even fatal. And so, the good news about our immune system is that we are beginning to figure out how to harness it and direct it to attack cancer cells. But the bad news about this immunologic approach to treating cancers is that it's very risky business, and that if the immune system, that we can go too far in activating the immune system, and it can cause serious or life-threatening or fatal complications to our patients. So I think those studies are promising. I think they have to be done very carefully and should only be done in the context of properly designed and controlled clinical trials.
Okay, great. Thank you. Great question. So the next question we have, how long does it take to manufacture the cells and get them back to the patient?
Yeah, that's a very good question. We did not talk about that. So at the moment, CAR T cells are not off the shelf. They have to be manufactured either on-site or sent to somewhere else to be manufactured. That process usually takes a minimum of 2 weeks. Different companies have different approaches in different manufacturing facilities. It can take up to 4 or 6 or 8 weeks. And so there is a period of time after the patient's T cells are collected through this apheresis process, but before the product is ready for infusion, where we have to keep their leukemia in check, usually with low-intensity chemotherapy so that patients don't develop lots of infectious complications. There are approaches. One of the new approaches that I didn't include in the slide for lack of time is that there are some investigators that are trying to make so-called off-the-shelf CAR T cells, so that we wouldn't use a patient's own T cells. We would have, if you would, a generic T cell with a CAR T cell already in it, so that a patient comes in, they're diagnosed with leukemia, and they could get their CAR T cells the same day, in theory. How well that's going to work is not yet clear. But that's another promising direction for research.
Great, thank you so much. So I think we have time for 1 or 2 more questions here. We're getting as many as we can in here. So the next one is a follow-up question to the first question regarding SCT. The same individual is asking, do we know with certainty that no patient treated with has required SCT?
No, I don't know that at all, because in fact, I suspect there are very likely patients who've received the CTL-019 product who have maybe attained a complete remission, but then lost their CAR T-cell product and subsequently had to go undergo bone marrow transplant. So there are no guarantees, absolutely no guarantees that this approach will help avoid bone marrow transplant. The practice now that has emerged is if patients receive a CAR T-cell product that has a possibility of persisting for long periods of time, we monitor the levels of normal healthy B cells in their blood, and if we see those normal healthy B cells that express CD19 starting to come up from very low levels, if the CAR T cells are there, we shouldn't be able to find any CD19 positive healthy blood cells in the body. If we start seeing CD19 positive normal lymphocytes turn up, then that's a signal, if you will, that the CAR T cells are going away or have gone, and then the patient's leukemia will inevitably come back without additional treatment, which could include a bone marrow transplant.
Okay, great. Thanks for that question. So we have time for one more question. I'm going to address the next question in the line. I'm sorry if we couldn't get to your question this time around. So the final question is, once patients are infused with the CAR T cells, how long does it take before you see an improvement?
Great question. So we typically wait 4 weeks before we perform a bone marrow to formally assess whether patients have had what level of response they had. If patients have circulating leukemia cells in the peripheral blood, we can often see those go away within just a matter of a few days.
Okay, fantastic. So once again, apologies if we couldn't get to your question this time around. And thank you so much to everyone for attending.